In this video, we're going to look at coat color in black labs. In the lecture notes, we looked at a cross between two black labs that were heterozygotes for both of the genes that control the coat color. Remember that we've got the B gene that's going to control the color of the pigment, and we're going to have the E gene that controls whether or not pigment is placed into the hair follicle. And so we're going to look at a cross between two black labs that are heterozygotes for both of these genes, as is shown up at the top of this slide. So first of all, if we're going to look at a cross, we have to look at how gametes are formed in one of these heterozygous black labs. And so here I've got a cell, and I'm drawing the chromosomes that are holding the B gene and the E gene in this black lab. So we've got one set of homologs. Remember, homologs are the same length, same shape, and carry the same genes, right? I've circled some homologous chromosomes here. And this first set of homologs carries the B gene, so the color gene. So the maternal chromosome has the dominant allele, the paternal chromosome has the recessive allele. And then the other pair of homologs carries the other gene, the pigment gene. And again, being a heterozygote, we've got one dominant allele on one chromosome and the recessive allele on the other. Now, if this particular cell is going to give rise to gametes, either sperm or egg for this organism, we've got to go through DNA replication first, right? So in S phase, the cell is going to take all of its chromosomes and it's going to duplicate them. So here I'm showing now the duplicated chromosomes. There's still only 4 chromosomes in this cell as I've drawn it, but they exist as duplicated chromosomes. Each chromosome has that pair of sister chromatids that are genetically completely identical to each other, so carrying the exact same allele for the B or the E gene. So now that we've gone through DNA replication, we can go through meiosis 1. Remember, in meiosis 1, the whole point here is to separate homologous chromosomes. And if we're going to separate those homologs, we've got to pair them up at the metaphase plate. Now, having this particular cell with 2 pairs of homologs, there are 2 ways that we can line up these homologous chromosomes at that metaphase plate. And I'm going to draw out both of the options. So option 1 is going to be on the left-hand side. Option 2 is going to be on the right-hand side. All right, so for option 1, here we're going to add both maternal red chromosomes on the same side of the metaphase plate. On option 2, we're going to add 1 maternal chromosome on either side of the metaphase plate. And then we'll draw in the paternal chromosomes. So the blue ones for option 1 are going to be both on the same side of the metaphase plate. And then for option 2, again, you're going to have 1 blue chromosome on either side of that metaphase plate. And I'm also going to draw in the alleles for both the B and the E genes so that you can see on which side those alleles happen to be on and how they're eventually going to be separated at the end of meiosis 1. And so essentially what I'm showing you here is Mendel's law of independent assortment, right? That pair of homologs that carries the B gene is going to sort itself independently at the metaphase plate from the pair of homologous chromosomes that carries the E gene. This gives us these 2 different options, option 1 and option 2, of how the chromosomes can sort themselves. All right, so we've got our chromosomes lined up at metaphase 1. We're ready to separate them and to go through the rest of meiosis 1 here to separate the homologs. So I've drawn all my my daughter cells, right? I'm showing you here is where we have our law of independent assortment taking place in meiosis 1, where the homologous chromosomes are sorting themselves independently at that metaphase plate. And now we can start sorting our homologs into the cells, the daughter cells at the end of meiosis 1. So for our option 1, one of the daughter cells is going to end up getting both maternal chromosomes, and so this particular daughter cell will get the dominant allele for the B gene and the recessive allele for the E gene. And then the other daughter cell is going to get both paternal chromosomes if those two homologous pairs of chromosomes had sorted themselves via option 1 in meiosis 1. For option 2 now, it's going to be a mix. So each daughter cell is going to end up with one maternal chromosome and one paternal chromosome. So I've drawn in the paternal ones in blue. Now I'm drawing in the maternal chromosomes in red. So in this case, the first daughter cell will get the dominant B allele and also the dominant E allele. And the other daughter cell will get both recessive alleles for genes B and E. And here's where we show the law of segregation, right? In meiosis 1, you separate the homologs. So one of the two alleles for the B gene goes into one daughter cell, and the other allele goes into the other daughter cell when you separate the homologs. Okay, so we're done with meiosis 1. But remember that the chromosomes are still duplicated, right? This is too much genetic information. So now we're going to follow through with our second cell division. We're going to follow through with meiosis 2. Here we're separating sister chromatids. So I'm going to draw out all of the potential resulting daughter cells. We've got 4 daughter cells that could result from option 1. And then the same thing is going to happen for option 2. We're going to have 4 daughter cells that could result from our second way of organizing the homologous chromosomes way back when in meiosis 1. And so now we'll separate the sister chromatids, making sure that each daughter cell, which will give rise to either a sperm or an egg, depending if this is a male or female lab. Each egg or sperm, each gamete, will get only one allele for each of the genes. So for option 1, we can have gametes that either have the dominant B allele and the recessive E allele, or the other 50% of the gametes, as I'm filling in here, will get the recessive B allele and the dominant E allele. And then for option 2, if we fill in how the sister chromatids are going to separate to give the resulting gametes, we are going to have then 50% of the gametes in option 2 are going to get 2 dominant alleles, so both the dominant B allele as well as the dominant E allele. And just in case you're not 100% certain what we're doing right now, right? I've drawn in the metaphase plate here showing you that we're separating those sister chromatids now in meiosis 2. And so the other 50% of the gametes in option 2 will get both recessive alleles. All right, so now what we've done is we've gone through meiosis 1 and meiosis 2 in these heterozygote black labs that have one dominant and one recessive allele for both of these genes, B and E, and we've shown how you can come up with every possible combination of gametes based on the law of independent assortment and then the law of segregation that we talked about when we were going through Mendelian genetics. So here are all of our gamete options: dominant B, recessive E, or recessive B dominant E, or both dominant alleles or both recessive alleles in the gametes. And so because we have 4 options for gametes, we have to come up with a 16 square Punnett square. And so I'm drawing it out here. And we're going to stick the male gametes, or the sperm, on the top of the Punnett square, and then the female gametes, or the eggs, on the left-hand side of the Punnett square. And so we'll fill in all the possible gamete options now. We'll start with the 2 dominant alleles. So you can have a dominant B and a dominant E sperm. You can have a dominant B and a dominant E egg. We'll do the same thing with the remaining 3 options for the gametes. And once that's complete, we start our crosses. And remember, when you fill in your crosses for a Punnett square, you always want to keep the 2 alleles for one gene together. So that means we're keeping the B's together and we're keeping the E's together. It is also proper to keep the dominant allele always first for a gene and the recessive allele second. And then the last thing we're going to do once I finish filling in this Punnett square is we're going to take a look at the actual phenotype. So remember, the E allele will regulate whether or not pigment is placed into the hair follicle. So those those Labrador retrievers that don't have a dominant E allele at all, they don't have the ability to place pigment into the hair follicle, period. Doesn't matter what color it was meant to be. And so you'll notice that in the bottom right-hand corner, we've got 4 labs that do not have a dominant E allele, and these are going to be our yellow labs because their hair follicles are essentially colorless. For the rest of them, they do have the ability to put pigment into the hair follicles. And so the B gene now will determine which color that fur pigment is going to be. So if you have 2 recessive B alleles, you're going to end up being brown. That's the color I just filled in there. And then the remaining labs all have at least 1 dominant B allele, indicating that they're going to end up having that black fur. And this should match exactly what you see in the book.